Now that we've taken a look at many of the different processes involved in innate immunity, all the way from alternate pathway of complement activation to acute inflammation, we're now going to zoom in a little bit on the job of the phagocytes and take a little bit closer look at the process of phagocytosis. Phagocytosis, of course, involves ingesting particulate debris, pathogens, and essentially helping to eliminate those from the host. This process, you can see in the image, involves a pseudopod that extends up to essentially trap either the pathogen or the debris and engulf it and enclose it into what's called a phagosome. There are a lot of really important events that happen in that phagosome that are super high yield, and we're going to talk about those in just a little bit. Eventually, that phagosome fuses with a lysosome, so now we have a phagolysosome. And once that happens, really, unless you have a pathogen with the capacity to escape a phagosome or a phagolysosome, which we do have, and those are super high yield, you're pretty much going to die within one of those vessels, either the phagosome or the phagolysosome. Once the pathogen or the debris is dead, it will essentially be digested, and eventually exocytosis will occur of the digested contents. What we're going to do is sort of talk in a little bit more detail as far as the process of phagocytosis. We'll talk about opsonization, and then we're going to really hone in on this process, dealing with both oxygen-dependent and oxygen-independent killing. We already mentioned sort of how cool the neutrophils are in this process of phagocytosis. Dr. Ali talked to you about acute inflammation, about the neutrophils sort of really rushing to the cause, dying there. Remember, they are not a prolonged defense against pathogens. They sort of act quickly and then they die, forming pus. I mentioned to you when we were talking about neutrophils in this chapter that even when the neutrophils die, they're sort of not done killing. They extrude their nuclear contents into the extracellular milieu, which form these neutrophil extracellular traps that can actually help to trap and kill pathogens. On top of that, you can also get some tissue damage, and this, of course, is going to be important not only in infectious diseases, but also in autoimmune diseases where you sort of have this chronic type of inflammation. You can actually get tissue damage from the neutrophils as well, when they sometimes will release their enzymes, the reactive oxygen species. Those get released into the tissues and can actually cause tissue damage. We already mentioned this process of opsonization. Opsonization is going to be really important at helping to eliminate extracellular pathogens through the use of both antibody and complement. Now, we have already mentioned that C3b is the major complement protein that is essential for opsonization, but you also need to know the isotype of antibody that helps with opsonization, and that's IgG. So you have to get really specific in knowing the molecules that can opsonize. That's going to be very high yield for step one. Okay. Now, what's really cool is you can have a macrophage that sort of binds to a pathogen through lots of the receptors that Dr. Ali talked to you about, and they can become activated that way. But this process of opsonization sort of makes things a lot easier for the macrophage. And if you have a pathogen that is coated with either complement C3b or IgG, essentially it delivers the pathogen right to the phagocyte. And this can actually enhance the process of phagocytosis, making it up to 4,000 times more efficient than it is without these opsonins. So antibody, specifically IgG, and complement, specifically C3b, are called opsonins, and they can enhance this process of phagocytosis that we just talked about through something called opsonization. Now let's take a look at what that entails. So as I just mentioned, the process of opsonization involves either complement C3b binding to a pathogen, as you can see in this image, or IgG antibody. And remember, this is specific for IgG and complement C3b because they have to specifically bind to receptors on the phagocyte. So C3b binds to a C3b receptor, and IgG binds to an Fc receptor that you also need to know as CD16. This is going to be an important CD molecule for you to know. We've talked about some CD molecules already that you need to sort of add to your arsenal. CD3 is obviously one of them. We talked about that one being a marker on all T cells. We talked about CD19 as a marker on all B cells. CD16 is important because it's the Fc receptor for specifically IgG. So we sometimes will call this an Fc gamma receptor because it specifically binds IgG. So now you can see the IgG and or the C3b bound to this pathogen and can deliver now this pathogen directly to a phagocyte, which makes it way easier for the phagocyte to engulf and kill that pathogen. So when we have deficiencies, and again, we bring this up because of course this is where USMLE is going to ask you questions. If you have complement deficiencies that affect C3b, or if you have humoral deficiencies that affect IgG, you're going to have a defect in opsonization, and that's going to manifest as recurrent bacterial infections, and that are often fairly severe. So keep that in mind because that's a frequent question they like to ask. Now we're going to talk a little bit about what happens during the process of phagocytosis that allows for these pathogens to be really efficiently eliminated during this process, unless, as we mentioned, a pathogen such as mycobacteria, for example, that has the capacity to escape this sort of killing. Now those pathogens are going to be really super high yield. They're going to ask you lots of questions about those. And it's important, and we'll just bring it up right now, that you understand that those pathogens that we call facultative intracellular pathogens need an entirely sort of different immune response to deal with them and help to try to eliminate them. And that actually is our type 4 hypersensitivity response that we will get to a little bit later. For now, we're going to talk a little bit more about intracellular killing. And in intracellular killing, what we are looking at is essentially two mechanisms that can help us eliminate these pathogens. We have our NADPH oxidase, which is also called an oxygen-dependent mechanism, that helps us essentially take molecular oxygen and convert it to all kinds of superoxide radicals, hydroxyl radicals, which are unstable and will degrade the pathogen. Will also degrade your cells if that happens to get released into the extracellular milieu. There's also myeloperoxidase, and myeloperoxidase I always sort of like to think of as the backup plan. The immune system, guys, has a ton of backup plans, just in case something doesn't go quite right. Myeloperoxidase, sort of the backup plan here, because if your NADPH oxidase is working, myeloperoxidase is sort of nice. It's a nice way to finish, but it's not completely essential. However, if the NADPH oxidase is not working, which we're going to talk about in just a little bit, myeloperoxidase is actually pretty handy to have around. Myeloperoxidase takes the hydrogen peroxide that can either be generated through NADPH oxidase or generated from the pathogen itself to essentially create bleach. And we know very well that there's not a whole lot of pathogens outside of maybe a prion that can actually escape killing in bleach. In addition to the reactive oxygen species we talked about, reactive nitrogen species are also important. These come into play from inducible nitric oxide synthetase, creating nitric oxide, which, among all of the different other things you know about nitric oxide's effects on the body, also has potent antimicrobial properties. Of lesser significance are the oxygen-independent killing mechanisms, which include things like lysozyme, which we already mentioned, which can degrade peptidoglycan from bacterial cell walls. Defensins, which we sort of mentioned already. They're not super important, but they can form channels in bacterial cell membranes. Lactoferrin, which essentially just steals iron, and some degradative enzymes that can help with the overall process of eliminating these pathogens. I can say with pretty good certainty that the oxygen-dependent killing mechanisms are way more important, even for, life in general, but on USMLE, because if we have deficiencies in the oxygen-dependent killing mechanisms, those manifest as very specific immunodeficiency diseases that we're going to come back and talk about time and time again, versus if we have deficiencies in these oxygen-independent mechanisms, we don't really see the effects of that. So let's just take a look at both oxygen-dependent and independent killing. If we look to the left, you can see those oxygen-independent killing mechanisms that we just mentioned. Again, sort of not as relevant as the oxygen-dependent killing mechanisms. We're going to sort of just walk through the oxygen-dependent killing mechanisms because, again, those are super high yield for USMLE. Essentially, you need to remember that NADPH oxidase is the enzyme that converts molecular oxygen to a lot of these superoxide and hydroxyl radicals. Those radicals are very unstable. They will degrade pathogens. They will also cause tissue damage. One thing I want you to pay attention to, in addition to those hydroxyl radicals, is the hydrogen peroxide. So that can be formed from NADPH oxidase, but also it's important for you to understand that bacteria can generate hydrogen peroxide without NADPH oxidase within the phagosome. So hold on to that little fact. We're going to come back to that in just a minute when we talk about chronic granulomatous disease. Okay? Now, we talked about the immune system being really good at having backup plans. So here's where myeloperoxidase comes in. Myeloperoxidase takes that hydrogen peroxide and essentially converts it to hypochlorite or bleach that can also kill a pathogen pretty readily. So the oxygen-dependent killing mechanisms that you see here are really sort of essential to this killing. And like I said before, and we're going to say it probably a million times because it's very high yield, that unless you have a pathogen that can escape these killing mechanisms in some way, shape, or form, like mycobacteria, listeria, legionella, or just to name a few, they are going to die within the phagosome. The last thing I'll just point out to you here at the bottom is the activation of inducible nitric oxide synthetase, which again creates the reactive nitrogen species as well, which also has antimicrobial properties. So keep this figure in mind, and now we're going to talk about what happens when we are lacking one of these very important enzymes that contribute to the oxygen-dependent killing. So as I mentioned, deficiencies in oxygen-dependent killing mechanisms manifest themselves as immunodeficiency diseases. And one of the really high yield and important diseases that they love to test on is chronic granulomatous disease. So we're going to talk about some of the features of CGD, and then we'll sort of walk through the pathway of CGD so that you really get a good feel for the types of questions that they can ask you based on this vignette. CGD is a deficiency in the production. It could be one of several subunits of NADPH oxidase. And essentially, this takes out Plan A, right? So Plan A of oxygen-dependent killing mechanisms is to generate the oxygen radicals and hydroxyl radicals that we talked about. And what happens when we don't have those oxygen radicals is we sort of rely on the backup plan. So remember, myeloperoxidase is our backup plan. And if the bacteria does not produce a catalase, then myeloperoxidase sort of takes the ball and runs with it, takes that hydrogen peroxide, converts it into hypochlorite, and can kill the pathogen. But where we have problems are with the catalase positive bacteria because they essentially destroy that hydrogen peroxide, eliminating that as a substrate for myeloperoxidase. What we see in chronic granulomatous disease are we see patients that have recurrent infections with catalase positive bacteria and fungi. So that's going to be one of the big clues, and I think why this is probably a pretty high yield question is that you have to obviously understand some immunology. You have to know your microbiology because they're not just going to, of course, say that, oh, you have this patient that's infected with catalase positive pathogens. They're going to list them. And you're going to have to know that the one thing they all have in common is that they're catalase positive. And then if you really want to stretch it, anytime there's an enzyme involved, the biochemists want to claim that as their own. So technically, you can write one question that encompasses biochemistry, immunology, and microbiology. Oh yeah, it's a done deal. So super high yield. So this is a figure we just looked at least one that looked similar to this earlier when we were talking about oxygen-dependent killing. But now we're going to walk through it in a patient with chronic granulomatous disease. So you can see that the NADPH oxidase is missing. So we are unable to generate these oxygen radicals that we talked about. We can see, though, that bacteria can generate hydrogen peroxide while in the phagosome. If they do, that's awesome, because myeloperoxidase can then use that hydrogen peroxide, convert it into hypochlorite, and kill a pathogen. So we can still get killing from our backup plan in patients with chronic granulomatous disease. But where is the issue? The issue is in pathogens that have a catalase enzyme, which essentially destroys that hydrogen peroxide, turns it into water and hydrogen ions, so that it cannot be used as a substrate for myeloperoxidase. So now, essentially, when you have a pathogen that's catalase positive, you already don't have plan A, that's your NADPH oxidase, and you are wiping out plan B, which is myeloperoxidase. So you wind up getting recurrent infections with catalase positive organisms. Now, one of the things that's also sort of important for you to understand is why on earth are they calling this chronic granulomatous disease? Why don't they call it chronic catalase positiveness disease? Because when a macrophage can't kill something, macrophages are super tough and they have a really strong work ethic. And if they can't kill something, they're going to do the next best thing, which is to attempt to seal it off in a granuloma. So these patients get recurrent infections, but they also get lots and lots of granulomas in an attempt to stop these infections. When we get to the chapter on immunodeficiency diseases, one of the things that we're going to have to do is differentiate between lots of different immunodeficiency diseases that could all look very similar, at least at the chief complaint. And the chief complaint essentially being recurrent bacterial infections. So that could arise from phagocyte deficiencies, from humoral deficiencies, and even from complement deficiencies. So we have to look at some clues that will sort of help us out along the way. One of the clues that they can put into those questions is something about an NBT test. Whether or not it's positive or negative can help you decide if you're going to say, okay, I'm answering this question as if it were CGD, or nope, I have to look elsewhere. So, in patients with chronic granulomatous disease, they are unable to essentially reduce the dinitroblue tetrazolium. And that is used as a test in patients to see whether or not their NADPH oxidase is working. The problem with this test is it's reported backwards, right? Like you don't have enough to know when we talk about this test is positive or negative. When they report this test as positive, that actually means that NADPH oxidase is there, it was working, and you did reduce that NBT. When they report this as negative, it actually means you're positive for the disease. So you just have to be a little bit careful in how they put the NBT test in there, but it really can be a good clue in a vignette. Now, NBT test is pretty simple to do. It's cheap, but there's another test that can be used as well, and that's the dihydrorhodamine test, and it is utilized through flow cytometry. So they may mention that as well. But oftentimes what we see on board exams is they'll sort of revert back to an older test to make you think a little bit more, especially we know how they like to write these questions. This test is a little bit confusing. So you have to remember when it's reported as positive, they don't have chronic granulomatous disease, and when it's reported as negative, they do. So a lot of the times they'll put that NBT test in there, so you just want to sort of be on top of it because it can really help you. Dr. Alley talked to you about the acute inflammatory response, and she mentioned the pro-inflammatory cytokines, which she already mentioned are extremely important and high yield, and those were IL-1, IL-6, and TNF-alpha. Those are produced during the acute inflammatory response, and she already talked to you about what some of their functions are. When you start to see the effects of those cytokines on the tissue, that shifts from an acute inflammatory response to a systemic inflammatory response. So when you have a patient that has a fever as the result of acute inflammation, your inflammatory response is now systemic. And we're going to take a look at a figure that I think will help you sort of delineate this response versus the acute inflammatory response. So, we mentioned acute inflammation. Dr. Alley went through that in that slide that she said could be a nightmare if you just sort of looked at that. We get the cytokines that we call pro-inflammatory cytokines, IL-1, TNF-alpha, and IL-6, and those have a systemic effect. And she already talked to you about these, but they can act on the hypothalamus to give us the production of fever, which we said wasn't always a bad thing. They can result in the production of acute phase proteins from the liver, such as CRP, and they can act on the bone marrow to help the bone marrow realize, you know what, we have an infection, we have to kick out some more cells, the system needs some help, and that's where we'll see the leukocytosis. So all of those are signs of a systemic inflammatory response that are a result of cytokines that were produced during acute inflammation. Those cytokines are going to be really high yield cytokines, particularly cytokines like TNF-alpha, where we have inhibitors or biologic molecules that inhibit those cytokines. That's one thing you definitely want to pay attention to. If there is a drug that can inhibit one of those cytokines, it becomes way more high yield for step one. Spent a lot of time now talking about the innate immune response to pathogens such as bacteria. We're now going to shift gears and look at the innate immune response to viruses, which consist of two major mechanisms. The interferons, which we're going to talk about here, and then the natural killer cells, which we are going to finish the chapter talking about. Interferons are a family of proteins that are classified according to the cells that make them. The interferon alpha and beta are produced by cells that are infected with a virus. And this is going to be just sort of a really cool response that you're going to see that these cells have to viral infections. They act on target cells to inhibit essentially protein synthesis. And what happens in the wake of that is that you inhibit viral replication. You are not specifically inhibiting the virus, and that's going to be an important distinction we want to make. So these aren't virus specific. It's causing essentially a temporary shutdown in protein synthesis. Interferon alpha and beta inhibit protein synthesis. Again, it's important for you to know it's not specifically inhibiting viral protein synthesis, but inhibiting protein synthesis in general. And it can do that by one of two mechanisms, either by activation of an RNase, essentially, which is going to chew up the messenger RNA, or phosphorylation of a protein kinase which inactivates EIF2, which also inhibits protein synthesis. So let's take a look at how production of interferons can protect cells against a viral infection. So we're first going to start on the left, looking at the infected cell. You can see that this cell is infected with a virus, and you can also see that this cell begins to produce interferon. Now, if you go all the way back to sort of what Dr. Ali talked to you about with toll-like receptors and some of the other important innate immune receptors, you'll understand again that some of those receptors become activated and can result in the production of these interferons. What's really cool is that a very potent inducer of interferon for cells is double-stranded RNA. And that actually makes perfect sense because we don't typically have double-stranded RNA. Viruses, though, of the bulk of which are RNA viruses, all at some point during their replication cycle will have to make double-stranded RNA. So it's a really good indication to the cell that something's there that shouldn't be, and it leads to the production of interferon. This cell that's infected produces the interferon, and it releases that interferon, which is going to bind to neighboring cells and initiate sort of this inhibition of protein synthesis. Okay? So remember, we're talking about activating either an RNase or something that's interfering with EIF2, but in any case, we are shutting down protein synthesis in that cell. So now this virus leaves the infected cell. It can still bind to and attach to the neighboring cells. It can actually still get into the cells and uncoat its nucleic acid, but that's where it stops. Because in order to make more virus, you need to be able to make viral proteins. And if the cell is temporarily shut down from making viral proteins, guess what? The virus isn't going to be able to make any proteins. So it's a temporary shutdown in the cell of protein synthesis so that when viruses attempt to replicate, they can't do so because they use all of our protein synthesis machinery to generate their proteins. Now, what's really cool is that once the threat of the virus is gone and there's no more interferon around, these cells can resume normal protein synthesis. So it's sort of just a temporary shutdown when we really need it and the threat of the viral infection is pretty high, and then we can sort of go back to normal homeostasis for those cells. We already mentioned that interferons were going to be really high yield. They're obviously high yield for the innate immune system, but they're also high yield because we can utilize them to treat various diseases, and we're going to talk about that here in just a minute. In general, though, the interferons, and again, we have to also include interferon gamma here, even though interferon gamma doesn't have a direct antiviral effect if we're comparing it to alpha and beta. But in general, when we talk about the interferons, they can increase the expression of both class 1 and class 2 molecules on the surface of cells. They can therefore then increase the efficiency of antigen presentation to both cytotoxic and T helper cells. So that's sort of part of their normal function. We have been able to utilize molecular biology and gene cloning so that we can produce these interferons and actually be able to use them to treat various disease states. Interferon alpha is really important as far as a treatment for a lot of different viral infections, one of which are the hepatitis viruses B and C. A lot of the times for treatment, when we're talking about hepatitis B and C, they'll often refer to as pegylated alpha interferon. But that, along with the use of other antiviral drugs, has actually been really successful in the treatment of hepatitis. As far as cancer therapy goes, interferon alpha has been showing promise in the treatment of various types of leukemias, such as hairy B-cell leukemia and CML, and even Kaposi sarcoma. Kaposi sarcoma, of course, is due to human herpes virus 8. So that makes perfect sense as to why interferon alpha might have some efficacy in treating that particular cancer. Interferon beta is really important, but this one's sort of interesting because interferon beta is used to treat patients that have multiple sclerosis. What's interesting about interferon beta is that we're really not sure the mechanism of how this works, but what's important is that in patients with MS, it does not clear up the plaques that are already there, but prevents the establishment of new plaques so that patients with MS can enjoy longer periods of remission. Interferon gamma is a cytokine, again, we're going to come back to time and time again because it's really important for macrophage activation, but it's a cytokine that we can use to treat chronic granulomatous disease. We mentioned the main function of interferon gamma is as a potent inducer of macrophage activation. And when we give patients with CGD interferon gamma, the macrophage can activate in such a way that it can sometimes compensate for the loss of the NADPH oxidase and the ability to generate those toxic oxygen radicals by upregulating everything else that the macrophage can do, like the reactive nitrogen species and the oxygen-independent molecules that can be helpful during these types of infections. All right, so natural killer cells are sort of the closer of the innate immune system chapter. So we're going to talk about some of the features of natural killer cells, and then we're going to talk about how natural killer cells function. Natural killer cells are completely innate as far as the immune response. If you look at their function, I'm hoping you're going to see that they're similar to another cell that is not considered part of the innate immune system, and those are the cytotoxic T cells or the CD8 positive T cells. They actually use a lot of the same ammunition for killing viral infected or transformed cells that cytotoxic cells do. The main difference, and this is a super important difference, is that they do not specifically recognize antigen as do the cytotoxic T cells. Natural killer cell activity is enhanced in the presence of not only interferons, but IL-12 that we talked about earlier. Recall from chapter 2 that natural killer cells develop from that common lymphoid progenitor cell with T cells and B cells. So they share a progenitor, but you don't want to ever get them confused with part of the adaptive immune system. They are not T cells. They do not develop in the thymus. They do not express CD3. Their specific markers that we're going to use to enumerate them or find them in the blood are CD16, which we already talked about as the Fc gamma receptor, and CD56, which is a cell adhesion molecule. They are not MHC restricted as the cytotoxic T cells are, and their activity does not generate immunologic memory as do the cytotoxic T cells. We're now going to talk about how natural killer cells kill. We're going to focus on their innate killing mechanisms in this chapter. In a subsequent chapter on cell-mediated immunity, we will focus on another killing mechanism called ADCC. But for now, we're going to take a look at the innate killing mechanisms of natural killer cells. Now, we already mentioned that the immune system likes to have backup plans. I like to sort of think of natural killer cells as a backup plan because essentially their function is very similar to the cytotoxic T cells. And cytotoxic T cells are perfectly capable of lysing and killing viral infected cells, especially in a very specific manner through their T cell receptor. However, when you have a cell infected with a virus, like CMV, for example, that can actually downregulate class 1 MHC expression, the cytotoxic T cell is sort of taken out of the game. That's where the natural killer cell comes in. Natural killer cells, through an inhibitory receptor, recognize whether or not a cell is expressing class 1 MHC or not, and if they are not expressing class 1 MHC, will essentially kill that cell because it knows that all nucleated cells should be expressing class 1 MHC. So what we're going to do is take a look at the receptors. We'll talk about the killer activating receptors and the inhibitory receptors, and then we'll talk about how they function. So the activating receptors, when they are engaged without the involvement of the inhibitory receptors, the target cell is killed. Okay? What that means is that the inhibitory receptor did not find a class 1-like molecule, and that means that the cell is likely either transformed or virally infected. If the inhibitory receptor as well as the activation receptor are ligated, then the target cell lives. So another way to sort of say that is that the inhibitory signal always trumps the activation signal. So let's talk about the killer activating receptors. There are a couple of different KARs expressed by natural killer cells. NKG2D is the major one, and I don't think necessarily USMLE is going to expect you to remember that as far as the name, but essentially what the activating receptors are doing. Okay? Now, the activation receptors have lots of different ligands. One of them are the MYC glycoproteins, which are actually stress proteins that get expressed only when cells are either usually infected or undergoing some sort of transformation. When the activating receptor binds to a MYC protein, the natural killer cells then unleash all of its ammunition, become cytotoxic, and can kill the target cell. The job of the inhibitory receptors is to recognize whether or not a cell is expressing class 1 MHC or not. So these inhibitory receptors, when they are ligated, actually shut the entire system down. And they do so by activating tyrosine phosphatases, which inhibit the intracellular signaling and activation by essentially stripping tyrosine residues from those signaling molecules. The inhibitory receptors on the natural killer cells bind to a very sort of specialized type of class 1 antigen called HLA-E. Now, Dr. Ali did not talk to you about HLA-E because this is not a traditional type of class 1 molecule, but you're going to see the very sort of unique and cool role that HLA-E has in the immune system and in natural killer cell killing. HLA-E is similar to the other class 1 molecules in that it has a ubiquitous distribution in the tissue, just like the other class 1 molecules do. And the HLA-E molecules actually bind to peptides that are derived from the leader sequences from your class 1 molecules, so HLA-A, B, and C. HLA-E requires that bound peptide for proper expression within a cell. So what you see is that the overall expression of HLA-E on a cell indicates whether or not the cell is expressing class 1 or not, or viral infected or not, or transformed or not. So the expression of HLA-E is sort of an indication to the natural killer cell as to whether or not that cell is expressing class 1 MHC. So it's a nice little sort of red flag or indicator molecule, and it also serves as the ligand for the inhibitory receptor. So let's take a look at how natural killer cells kill using their innate immune defenses. So we're going to take a look on the left. Natural killer cells express, we already talked about, two receptors, an activating receptor and an inhibitory receptor. The activating receptor recognizes stress molecules like the MYC proteins that are produced when cells are sort of in trouble. And the inhibitory receptor, we said, recognizes the expression of HLA-E as sort of an overall indication as to the health and well-being of the cell. If both the activating receptor and the inhibitory receptor are ligated, the natural killer cell won't kill that cell. Now, you might be saying, well, what if it's infected with a virus and it can't tell? Doesn't matter, because even if somehow this cell is infected with a virus, okay, it knows that it's still expressing class 1 MHC. So this cell isn't going to kill it. It's going to allow the cytotoxic cell to do it because it knows that it does that with sort of more specificity. So now let's take a look at the right. You can see that the activating receptor has still ligated the stress molecules on the target cell, but the inhibitory receptor has not identified the expression of HLA-E. When HLA-E is not there, remember the cell is not doing well. It could be transformed, it could be virally infected, and that is an indication then to the natural killer cell that A, the cytotoxic T cell can't kill this because it's not expressing class 1 MHC, so I'm going to have to pick up the slack and kill this target cell. Okay? Again, completely innate. And we're utilizing, essentially, the expression of class 1 MHC as an indication, really, as to whether or not the natural killer cell is going to have to lyse and kill these target cells. Now, what's really kind of neat when we talk about natural killer cells, and we've already sort of alluded to this other mechanism of killing through ADCC, we just looked at the innate killing of target cells by natural killer cells, so we know about the utilization of the activating receptor and the inhibitory receptor. But when natural killer cells are activated through CD16 or the FC receptor, we only actually need one signal required because the antibody being there signals that there's an active infection. And this is the mechanism that we're going to talk about a little bit later in Chapter 8, called antibody-dependent cell-mediated cytotoxicity, or ADCC. It's a really good example of collaboration between the adaptive immune system and the innate immune system. This table actually sort of summarizes for you the cytokines that are important in the innate immune response. We have an entire table that's in the appendix of all of the different cytokines. And so we don't want you to think that these are the only ones that are important, but it is important to know the innate functions of these cytokines because those tend to be a little bit more high yield. So right up front, you can see here that we have the pro-inflammatory cytokines. You can see sort of what the target tissues are and what the activity is. Here you can see a lot of the cytokines that Dr. Ali talked to you about. IL-8, which we know is super high yield as a chemotactic molecule. IL-12 we've talked about as far as a molecule that's going to be important in the development of cell-mediated immunity. And IL-15 and 18 also have target tissues, including the natural killer cells, that can help with not only proliferation, but interferon gamma synthesis. We haven't really talked much about IL-10 here, but it can be produced by macrophages and dendritic cells. We're going to come back to this one, talk a little bit more about it when we get into the chapter dealing with the T helper cell subsets. And last but not least, the interferons. It's important for you to know what the function of interferon alpha and beta are, especially, again, because they're used as therapies for different types of diseases. TGF-beta is going to be a really important cytokine. It has anti-inflammatory properties, but we're going to come back to TGF-beta in one of the later chapters when we start dealing, again, with cytokines that are important for class switching. So we teased out some of the cytokines important in innate immunity, but again, these aren't the only ones that you need to know. The bigger table is going to be in the appendix for you, so you have sort of a summary of all of the different functions of all of the different high-yield cytokines.